Welcome to another lecture by MedicoMedics, Cell Biology, Chapter 7, Genetics and Molecular Biology. In this lecture, we will discuss the structure of DNA, talk about DNA as a genetic blueprint, replication. We will discuss transcription, making RNA, and translation, making proteins. Briefly talk about regulation of gene expression, mutations and their effects, a brief overview of epigenetics, and end with a summary. Now, what is the structure of DNA? It is made of a double helix, so two strands twisted into spiral shape. You can see that clearly in this illustration. So we have one here and the other one here. Now, what are the building blocks? Well, we call them nucleotides. These have a phosphate group. They are made of sugar or deoxyribose, nitrogenous bases or nitrogenous bases. So adenine, thymine, cytosine, and guanine. We have a base pairing rule when we talk about the structure of DNA. So A pairs with T, meaning adenine pairs with thymine. Cytosine pairs with guanine, and they are held together by hydrogen bonds. So here is an illustration of a normal DNA sequence. Again, cytosine binds with guanine and adenine with thymine. And here on the right, you have the nitrogenous base illustrated here and here. The minor groove of the helix is here. The major groove is found here. And then we have the sugar phosphate backbone. So the phosphate and the sugar molecules form the structural framework of the DNA, while the nitrogenous bases store and transmit genetic information through specific pairings. This interplay of structure and function allows DNA to serve as a stable yet versatile molecule for encoding life. So DNA as a genetic blueprint. We have genes that are segments of DNA that code for proteins. And we have another term, which is genome, which is the entire set of DNA in an organism. Now, the function is that DNA stores, transmits, and expresses genetic information. Proteins that are made from these genes determine our traits, like eye color or our blood type. So DNA is like a recipe book, with each gene being a specific recipe for a protein. Now let's explore how DNA is copied during cell division to ensure genetic continuity. So DNA replication, the purpose is to produce identical copies of DNA for daughter cells during cell division. The steps include unwinding, so DNA strands separate. Base pairing is step 2, so new nucleotides pair with exposed bases. And then we have sealing, so DNA polymerase forms two identical DNA molecules. So if we do a step-by-step brief explanation of DNA replication, we start with the unwinding of the DNA. So helicase, the enzyme, unzips the DNA by breaking the hydrogen bonds between base pairs, creating a replication fork. We then have single-stranded binding proteins, or SSBS, that stabilize these unwound strands. In step 2 then, so we have the unwinding, which was step 1. We then enter step 2, so RNA primer formation. So the enzyme primase lays down an RNA primer. It's a short RNA sequence to provide a starting point for DNA polymerase. Further down, we have leading strand synthesis. So the DNA polymerase now adds nucleotides continuously in the 5 to 3 direction, following the replication fork. And the replication fork was created in step 1. We then have step 4, lagging strand synthesis. Right? On the lagging strand, which runs 3 to 5, DNA polymerase synthesizes Okazaki fragments discontinuously in the 5 to 3 direction, each starting from an RNA primer. After that, we enter step 5. So we have primer removal and replacement. So RNA primers are removed by DNA polymerase 1 or similar enzymes and are replaced with DNA nucleotides. And then finally, we arrive at step 6, which is sealing the gaps. Remember here, sealing the gaps, where DNA ligase joins the Okazaki fragments on the lagging strand, sealing the sugar phosphate backbone. Now, DNA is not the final product. Let's see how cells use DNA to make proteins. Now, here is something we call the central dogma of molecular biology. So the flow of genetic information is DNA to RNA to proteins. Also illustrated here, we have the DNA here, we arrive at RNA and then to proteins. And this is the end result, the thing we want. Now we have two very high yield and important terms here for you to pick up on. One is transcription. That's when DNA is copied into RNA. So from this step to this step is transcription. We then have the second one, translation, where RNA directs the synthesis of proteins. Now, important here is proteins carry out most cellular functions, like enzymes, hormones, and structural components. So proteins are indeed important. So to give you an idea or a metaphor, think of DNA as the master plan, right? And RNA as the messenger of this master plan. It would then make the proteins as the workers building the cell's structures. Now let's look at transcription, which is the first step of this central dogma. Now, transcription, the making of RNA. The purpose of transcription is to create a single-stranded RNA copy of DNA. Steps include initiation, so RNA polymerase, the enzyme, binds to DNA at the promoter region. The second step is elongation, so RNA polymerase adds complementary RNA bases. And finally, we have termination, so RNA polymerase stops at a termination signal, releasing the RNA. Now we talk about different types of RNA. We have mRNA, which is messenger RNA. Messenger RNA carries the genetic code. We have tRNA, so transfer RNA. These help in assembling proteins. And then we have rRNA, or ribosomal RNA, which forms the ribosomes. And if you remember, ribosomes are protein factories. So transcription is like copying a recipe from a cookbook. So the book, right, the cookbook with all the recipes is DNA. And transcription is like copying this onto a piece of paper and taking this piece of paper into the kitchen. This is transcription. The next step then is translation, where RNA instructions now are used to start cooking proteins. So translation, making proteins. The purpose? To convert messenger RNA into a sequence of amino acids, meaning proteins. The steps here are the same. So we have initiation. mRNA binds to a ribosome. And the first tRNA or transfer RNA pairs with the start codon. Then we have elongation. The ribosome reads the messenger RNA codons and transfer RNA brings amino acids to build the protein. And then finally we have termination. The process now stops at a stop codon and the protein gets released. And we also have some regulation of gene expression. So why is regulation important? Well, it ensures proteins are only made when needed. Some mechanisms of regulation include transcriptional control, so turning genes on or off, post-transcriptional control, modifying RNA, post-translational control, activating or deactivating proteins. So gene regulation is like controlling a music playlist, playing the right song at the right time and then skipping unnecessary tracks. Next, let's explore what happens when the DNA sequence is altered. So let's talk about mutations and their effects. A mutation is defined as a change in the DNA sequence. So we have different types of mutations, like point mutation, where there is a change in a single nucleotide. Insertion and deletion, so adding or removing nucleotides. We have chromosomal mutations, large-scale changes like duplications or deletions. We also have different effects. So a silent mutation has no effect on proteins. Missense mutations change one amino acid, for example, sickle cell anemia. We have nonsense mutations, so it creates a stop codon leading to a shortened protein. If you remember, the stop codon needed during termination was when the protein was let go to go off and do its function. But if it creates a stop codon, it may make it shorter than it's supposed to be. Then we talk about nonsense mutations. And here you can see a normal DNA sequence, everything is in order, and we have the production of normal proteins. If it's an abnormal DNA sequence, we will have an abnormal protein. Now, what is epigenetics? Changes in gene expression without altering the DNA sequence. Mechanisms include DNA methylation, so we add a methyl group to DNA to silence genes. Another way to do it is histone modification, so loosening or tightening DNA packaging to regulate access. This explains how the same DNA can produce different cell types, and it has big implications for diseases and inheritance. So epigenetics is like adding bookmarks to our cookbook, right? Highlighting some of the recipes and hiding others. So in summary then, DNA is the blueprint of life, storing genetic instructions. The central dogma explains the flow of genetic information, DNA to RNA to proteins. Gene expression is tightly regulated and mutations can alter cellular function. Epigenetics adds another layer of control over how genes are expressed. We have now reached the end of this lecture. Continue now to Chapter 8.